Hello, everybody. It's Gareth. This is the Somewhere on Earth podcast for Tuesday, the 24th of September, 2024. We're in London and we have guests, well, from the United States, as you'll be hearing. And also, as you'll be hearing, with us today is Anya Diktorovich, producer and editor of this podcast, but also wearing your expert commentator hat, aren't you, Anya, with a story that well, basically, we're going quite big on this one, aren't we? We're doing a single issue edition.
We are, and I wouldn't say I'm an expert commentator because it is quite complex, and I think I'm going to be asking quite a few questions rather than giving any
we'll qualify that.
Yeah, I think,
Should we say presenter's friend?
Yeah, just say presenter's friend. Our other experts are studio experts, but I'm definitely not one in this one. But this is the kind of thing that, I saw it and I thought, ooh, that looks interesting. And then the more and more I read, I was like, ooh, this actually looks really interesting. And then I kind of thought to myself, ooh, I wonder if this is going to win maybe kind of a big science prize at some point. If this technology works, I really think it could be the one that triggered it, this bit of research, the one that will be remembered for important prizes like, dare I say it, Nobels. I mean, I read it and I just thought, oh my goodness me.
Right, yeah. Well, you've been a science journalist for a long time. You've been
Quite
around the block a few times on the journalism beat. So, I'm taking your enthusiasm seriously, and it'll be really interesting to see how this pans out. So, Anya, you've teased that brilliantly. We haven't said a word.
This is a podcast, so if there's something you kind of may not understand completely, just press the rewind button, because I think some of it is, for me, it took a while to get it into my head and to process it. So that would just be my little kind of bit of advice for this one.
A bit complex. Or, dear listener, just reassure yourself with the knowledge that however little you know about this, I know even less about it, believe you me. So you will probably hear me during the interview quite often stopping and giving my idiot's version of what I think they're trying to do to see if they agree with my assertions. So, dear listener, between you and me and Anya and our guests, we'll work our way through it, and we'll certainly tell you why we think it's important and why we think it matters. You'll certainly pick that up, and don't worry too much if you don't get all the finer scientific details. There we are. Reassuring note over. I think we should jump in. And coming up today, we're devoting the whole show, really, when it comes down to it, to the future of computing. Well, okay, let's qualify that. It's one possible technology, perhaps, for the data centres of tomorrow. It won't necessarily replace what we have now, but it may have the potential to store all of the world's information in something the size of just a few cubic centimetres. We're excited because even though humankind, of course, has seen massive strides in computing since, say, around the 1960s, the underlying technology has been basically the same, based on silicon. But today, as we'll hear, we're kind of ripping up the textbooks on which my electronics degree was based. Yes, we really are. Stay tuned. It's all right here on the Somewhere on Earth podcast. It's not every day that we get to say this, but a whole new way of computing could be on its way. Not this year or next year, but possibly within about the next 5 years. So we're not talking like centuries off. This is relatively soon-ish. But don't hold us to that, obviously. Now, I can sum the gist of it up in just 3 letters. DNA. Yes, the molecules in all our cells, DNA, that code for everything that makes us, or indeed just about any other living thing. DNA is nature's information store, so why not use it as our data store in computing? In fact, the idea isn't new, but the research up for discussion today is using DNA not just for storing data, but for retrieving, computing, accessing, erasing, and rewriting data as well. That's what teams from North Carolina State University and Johns Hopkins University in the United States claim to have achieved in a world first. They call it a primordial DNA store and compute engine. And even now, it can apparently solve simple problems like simple Sudoku and chess problems. Well, let's meet the researchers. First, Orlin Velev, S. Frank and Doris Culberson Distinguished Professor of Chemical and Biomolecular Engineering at North Carolina State University. Orlin, what a title. Welcome along. So, we're going to just try and pick our way through all this. But your contribution to the research has been like some kind of unique material on which this is all based. This fragile, well, kind of not fragile, but kind of fragile stuff called DNA, this molecule. But it needs a material to sit on, doesn't it, in order to do what you need it to do in this bit of science. So tell me about that.
Our contribution to this work is providing a material that can basically protect DNA for a while it has been stored, and that allows us to manipulate DNA in order to keep it protected and then move it around and maybe extract the information further. So basically we're the material scientists in the team. And the real important aspect here is the DNA information storage, which has been provided by my colleague Albert's work.
All right, well, so we'll get into that then. So your key materials aspects to this then, as you say, it is the creating a structure in which the DNA can do its thing, because DNA is a remarkable molecule. Of course, it contains the instruction kits that code for ultimately living things. But is your point that DNA is actually quite difficult to work with, and so you need to just scaffold it almost into some kind of material where it's stable enough for you to use it in some meaningful way? And that's the bit you've done, as I understand it. Is that right?
My outlook is basically that DNA is actually a pretty resilient molecule on its own for a while, but it needs to be stored, it needs to be protected if one really wants to achieve long-term storage of information. And it's okay that we developed a material, invented a material that can do the job very well. And in addition to that, it allows to add the magnetic component that then we use to manipulate the DNA soft dendritic coils. This is our material complex. So in what we have been discussing with people is basically if the DNA is the hard disk, we're providing the box and the handle of the hard disks, so one can protect the information and one can move it around by using our new material.
Oh, yeah, we can run with that analogy. Well, definitely all roads point to Albert. This is Albert Kyung, who's also another author on this paper, associate professor, university faculty scholar, and Goodnight Distinguished Scholar, also at North Carolina State University. Welcome along, Albert. We've been talking about you behind your back there, haven't we? So you are the guy who can tell us a little bit about the computational element here then. So I did very badly, by the way, in biology at school. So treat me like a beginner. But even I know that DNA carries these four letters, doesn't it? You know, the A, T, the C, and the G of DNA that are the instruction code for life. I hope I put it okay. Yeah, so you're kind of working with this stuff. Pick it up for me.
So DNA is ACTs and Gs, and I like to, tell my students that the human genome is 3 billion ACTs and Gs, and if you convert the ACT and G into zeros and ones, so for example, A is a 0, 0, and a T could be a 0, 1, then you can essentially think about convert your genome into an amount of digital information, and that equals about 1 gigabyte. So the human genome is about a gigabyte of data, about like a CD-ROM or a DVD. And why we're really interested in DNA as a information substrate or storage material, computational material, is because that 1 gigabyte is just one of the 10 trillion cells or so in your body. And so if you take 1 gigabyte multiplied by the 10 trillion cells in your body, that means that you're literally walking around with more than all of the world's information in just the volume of your human body.
Oh, I love that. Wow. Okay. And I love the way that you also said that you can think of, say, the letter A. I'm being arbitrary here. You might tell me I've got the wrong letter, but, the letter A could be 0, 0. The T could be the 0, 1. The C could be 1, 0. So all I'm leading up to is that the entire computational revolution that's made all this stuff happen, that's enabling this conversation and for people to hear this conversation and all the other computing around it, is based on binary, isn't it? It's like ones and zeros. But the incredible thing about DNA is it's quaternary. So each bit of information is made up of potentially 4 options, whereas binary, of course, is either on or off. It's 1 or 0. And that's how you can sit there and just calmly tell me that our bodies can code like more than the sum total of human knowledge and all the data in the world walking around.
It also is a great illustration of how, we really are excited about thinking outside of the box and thinking about how information might actually exist and be computed upon in very different ways than we're used to. I think we're very much a binary focused, on-off, logic-gated thought process when we think about computation and information. But biology doesn't work that way at all. There's a lot of kind of in-between type of calculations, gray zones that are semi-on or semi-off, kind of analog, if you might put it that way. But many other types of analogies of ways that you might think about using biology or computing with biology or storing information that is just very different. And that really is exciting because it opens the door for new potential capabilities that we don't have with electronic media.
Yeah, I can't wait to hear about those uses and capabilities. But before we get into all that, Anya, let's bring you in because you're very keen on doing this story and you were the one saying, look, let's go big on this one. So what is it that's kind of lit up for you in this story?
I remember learning about DNA and just how exciting it was when I was like 16, 17, knowing that these little tiny building blocks that match up in pairs that then kind of form this twisted ladder that folds in on itself and folds in on itself and then wraps itself around histones, which are like these little balls of protein, I believe. I might be wrong. I may have forgotten all my biology. And it folds and it folds and it folds and it folds and it folds. And we have just under 2 meters worth of DNA in every cell in our body. And it's all there. All the information is there. And to kind of think that we can now use that instead of this 0-1 technology that we've been using all the time and say, look, guys, there's Orlin. He's developed this amazing sticky structure that seems to be able to have this coded DNA. Albert's there saying, oh, we can store this data here on the DNA. Oh, but we can also do some clever things with it. We can retrieve it. We can do some computational things there too. I just was like, oh, this is good. And every time I read another bit, I kind of sat back and thought, oh, that's clever as well. And oh, that's clever as well. But seeing that I'm not that clever when it comes to this, I suggest we get Albert and Orlin to kind of come back and talk to us. Because Albert, you've been working on this for a while now, haven't you? Since about 2016, and it was Orlin's work
right.
that really
and I think Orlin's a very gracious scientist, and I think a model for the type of scientists that you want to be, pioneering work with and pioneering work for a society. I think the past 6-7 years that we've been, or I guess 8 years that we've been working on this problem, there's a lot of things that we can do, and that the field in general has done a lot of brilliant work on thinking about how to, encode data, images, text, movies into ACTs and Gs. A lot of work on how to store it stably over long periods of time. And then, interestingly, in a pretty disparate field, there's been work on how do you do computations with molecule. Actually, a lot of that work inspired the system that we use to do computation specifically. The challenge, I think, has been, and that really being able to work with Orlin solved, was that we want to do something practical, right? Ultimately, that's what the field of both storage and computation with DNA wants to do. We want to create something that is practical, that is scalable, that the lay public will hear about it and be like, oh, that actually sounds like a computer. And so the challenge has been that you need to not only store the data and compute upon it, but you need everything in between. You need to be able to access that data after you store it. You need to do it repeatedly. You need to be able to erase data and add new data, right? You need something that is end-to-end, that's compatible with the whole process. And so that's what the material that Orland's lab has been working with and pioneered has really unlocked.
And it's a kind of sticky material then, isn't it, Orlin? Because I like the way that you said, look, that over on Albert's side of the research, they're the people coming up and designing the hard drive. We're designing the box that the hard drive sits in.
Right. I think that the cooperation that we established with Albert's group is really an example of how multidisciplinary research can deliver new areas and new scientific directions these days. Indeed, our material has been a new type of particles that are very sticky. And when I say sticky, that's kind of a little bit more special, because sometimes stickiness, I mean, you can associate it with gooey material, if you like, or some kind of glue. In this case, it's all physical. It is based on van der Waals interactions. There is this general similarity to the legs of gecko lizards, which can climb on any surface. So we found out that these materials can be great ways to compartmentalize Albert's DNA material, so it can be then stored, moved around, combined with microfluidic devices where one can use small volumes of liquid in order to store the information. So we contributed the material, and really Albert has been the pioneer of the information side. So hopefully our material would enable better storage of information, better handling, being able to really retrieve the right amount of, the right type of amount of DNA. And that's why we're very glad to collaborate on this project.
The way I understood it, silicon has an anchor, magnetic tape has an anchor, and Orlin, what you've done is you've given this free-floating DNA that's got this data stored in it, you've given that an anchor too. So suddenly your DNA is just like these other data storage devices, isn't it?
I think that it's a little bit hard to compare the approach here to silicon, because you really have a liquid-based bulk of the liquid type of process. Previously, people have been able to manipulate DNA by attaching it to particles and having it on the surface of particles. However, this is not a very good way to protect the DNA because it can go outside. So now if you have a porous particle where the DNA can hide inside, you can think of this as a tree where it kind of goes and is being immobilized in the tree. This can be a better way to protect it and a way to store more DNA per particle. So that's basically our contribution. And from there on is really the information processing that is opened by storing information in this biomolecule, and it's really amazing.
Presumably at some point, though, it needs to interface with, as it were, conventional computing materials, because you need to get the data into it somehow. I'm just throwing this around. Do you mount it on a chip surface or something like that so that you can, at the very least, upload an instruction set so it can even start to do any computing?
From our perspective, we really have a particle, so it's all in the bulk of a liquid, so we have a particle that can move together with the DNA around. So it is really not so much even the ability to mount it, but the ability to capture it and move it around. And then Albert can comment now how this enables the readout and the manipulation of the DNA.
And that's just where I wanted to pick things up, on the readout aspect there. So, Albert, just to help me out with this then. So, when you do some kind of, presumably quite simple at this stage, compute, what pops out the other end? Do you have like a great big plasma screen in the lab with a couple of numbers on it? Or is it a couple of molecules that turn a different color and you read them off on a microscope? What are you actually looking at?
That's a great question, and it requires 4 different research groups to do this. So the system basically looks like little tubes. They're microtubing, so sub-millimeter diameter tubing essentially, and we have our data stored in it on these dendritic particles that Orland's group made. And we use enzymes to basically copy the data into the DNA data into RNA, and then all that microfluidics really relied on expertise from Adriana San Miguel's group, also at NC State. And that allows us to do all of this in an automated, low volume, fast way. And then once we have that copy of the information that we want, those RNA molecules can be put through what's called a nanopore sequencer. And so this is where Winston Timp's group at Johns Hopkins University, they're experts on nanopore technology and sequencing and genomics. And what that does is it actually gives you a live readout of the sequences that are coming through. And of course, backending all of this is a encoding mechanism and decoding mechanism that's computational, that's really computer science, where you have to turn those strings of ACTs and Gs into images or text. And so that's where James Tuck's group are the experts in that. And what you basically come out on the other end, what you see is images of Drosophila embryo, of cells, of airplanes. You can also put text for poems or movies. So basically what goes in is DNA. What comes out, strings of ACTs and Gs that we convert into digital files that you think about.
Right. Okay. So, and quite a lot to unpack in that answer, because I did mention treat me like a beginner here. So here goes. Right. So you're talking about DNA, RNA, nanopore sequencing, and what have you. So I think to help me and others out is to not assume that you have something that looks like a display that I'm looking at now on my laptop. It's very different to that. You say it's DNA in, DNA out. In other words, what we're getting to here is that this is all happening in DNA, and then it goes into a sort of companion molecule, if you like, called RNA. But it's some kind of molecule that pops out the other end. I'm making it sound much easier than it is. And nanopore sequencing, there are lots of different forms of sequencing, but sequencing is when you read off these letters, isn't it? That's what sequencing is. There are lots of different ways of doing it. You've just happened to mention one called nanopore sequencing, and it's through that you see some kind of molecule effectively, and the answer's coded into that molecule. Am I even vaguely right? How am I going to do on my RNA nanopore sequencing exam here? Anywhere near?
Yeah, that's great. Yeah, that's great. It's perfect. You start with the DNA that's bound to this tree-like particle, this what we call the dendritic particle. We use enzymes. They're basically molecular machines that can make copies of that into RNA, which is a different type of biological molecule. And the nanopore sequencing is exactly how what you said, which is you need to figure out a way. You have a pool of millions of RNA molecules, and you need to know the sequence of letters of each of those molecules in order to get your data. And what the nanopore sequencer does is it feeds each molecule through a nanopore, a very, very small pore. And through electrical sensing, it can detect whether an A is passing through the pore, and then a T, and then a C, and then a G, and it can give you that kind of electrical signal that corresponds to the letters in your molecule.
Right. Now, I'm not trying to be horrible here, but if I want something, my laptop to do something, I type something in, it does it. Boom, there it is on the screen. You're making it sound very difficult here. What I'm fishing around for is this compute with potentially enormous power, but maybe a bit slow?
Yeah, that's right. So it is not going to be doing, at least, we haven't envisioned yet it doing what your computer, your laptop can do. Or even what, an analog calculator based on, for those of you that grew up in the era of analog calculators, it can't do that either, right? It won't be giving you an immediate response. It will take, right now, it'll probably take several hours to get, to do a computation and get the results out. So the application space, at least right now in our imaginations, and the limits of our imagination, is not the laptop computer.
So what is it going to be then? Because I was so excited until you said, by the way, this takes ages.
Yeah, so what we're really looking at is large-scale storage and computation that eat up a huge amount of land, water usage, cooling, energy, and materials, mining, and that have very outsized impact on economies, on environment, on climate. So these are things like huge data centers, of which up to 60% of that data is actually not accessed. It's called cold, meaning it's written onto a storage media and then saved, but not used. but it still has to be cooled, put into a data center, housed, and every several years it actually has to be copied over to a new storage media because a lot of the conventional technologies, these materials degrade over time. And then there's also applications when you hear of the AI revolution and machine learning, artificial intelligence, you also hear about how much energy these processes consume. And so, I mean, one way to think about it is the most natural intelligence, our brain, consumes 100 watts of energy, but we do really complex things. And so DNA has that type of potential to do these very complex, very challenging computations that even with the most powerful computers right now take days to run. We envision that we can do those types of applications using DNA.
Right, Anya.
When I first spoke to Albert, I just kept thinking back to the program that we did on data centers and water usage. So a huge drought in Chile will be going on for about 40 years, and the government's planning many, many more data centers. So we're not only talking about huge energy use, but also water use and the environmental impact. And Albert, the DNA, it's so compacted, isn't it, that you can actually store all the world's information in just a few cubic centimeters, potentially. Would we be looking at maybe getting rid of data centers altogether?
I think that there will be an increasing number of technologies that can specialize and address different needs. I think that DNA could take a huge chunk out of data centers, but there will be things that need silicon-based processing that's rapid, right? So email servers and things that require fast responses, those will stay probably on silicon-based computing. But things that like old photos or even, photos that you took last year, but you don't really want to access all the time. A lot of the AI modeling requires storing telemetry data of the calculations that they're doing, basically histories of their computations. And these are generating massive data sets. All of those we envision trying to replace. And so cutting down the footprint of data centers, but not eliminating them.
Yeah, absolutely. I could just imagine how much it would cut if I could store my photos on my phone. That would certainly help the speed of my calculations and processes on my phone. But also, what about things like transport costs as well? Because if you're having to transport huge servers here and there, that's obviously a major impact with data servers. I'm assuming, again, because everything's so small, the whole concept of data centers, at least the DNA storage ones, would be completely different to what they are now. How do you envisage they would look? What would be their footprint on land?
Yeah, I think it's really interesting that you mention kind of transport, because I think I certainly did not know this before I started this work in this area. And so I think a lot of people might not realize that if you need to transport or copy data from the West Coast to the East Coast of the United States, for example, or from New York to London, or Earth to the Moon or to Mars, right? Transmitting something like a petabyte of data, which is not actually all that much. It's 1,000 terabytes, so maybe 1,000 times the size of your laptop hard drive. The amount of time it takes to transmit that data can be something on the order of days, if not longer. And if you have like a company or enterprise scale data set that you need to copy over, that can take forever. It could take a year. And so actually what people often do is they physically load servers onto trucks and they drive them across the country. And that is faster than sending it over cables and the internet.
Wow.
Yeah. And so DNA, you can envision putting it into a FedEx box, like an envelope, and just mailing it.
That's a thought. That's mad that people just go and put a whole load of hard drives on the back of a truck rather than do it through the internet. Oh, the things you learn on this podcast. Alrighty. Just before we wrap it up by that way, then, for one thing, we're not saying that this is going to bring the end of silicon computing. It sounds as if it's going to be a complement or complementary technology rather than just chucking away everything we've ever known about computers. And clearly, I'm not going to have this DNA computing going on in my laptop. It's just like completely different, and I do get that. But I suppose what I am just wanting to finish with is, when do you think this might happen? You're doing some stuff in the lab at the moment, but any chance of this being commercialized or becoming a reality anytime soon?
I would not be surprised if in the next 5 years there were DNA storage applications for kind of long-term cold archival storage of data. I think probably things like warmer data processes, things where you're accessing the data every day or two, things where you're computing. I think those are more likely to be a decade or more out, at least. But I would not be surprised if we see long-term storage or kind of archival storage solutions popping up in the next several years.
All right. Well, thank you very much indeed for telling us all about this brand new research that has come out, and potentially this future really of DNA and RNA based computing and storage. Albert Kyung, we appreciate it.
Thank you. Thanks for having us.
Thank you. Associate Professor at North Carolina State University, from where we've also just been hearing from Orlin Velev. Thank you to you both, and to you, Anya. And of course, there's more in the podcast extra, in the subscription version of this podcast. But if you're not subscribing, then you might want to consider doing so. Just $10 a month. Otherwise, we will see you this time next week. That's a very radio thing to say, isn't it? Because with podcasts are a bit timeless. But you know what I mean, folks. We will appear in your podcatcher in a week's time with yet more technology goodness. But for now, from all of us, from production manager Liz Tui, producer, editor, and as you've been hearing there, experting, Anya Lutherovich, and me, Gareth Mitchell. Thanks for listening. See you soon. Bye-bye.